Okay, so our next speaker is Lawrence Reiter from the University of Texas Health Science Center. He's going to speak with oh, sorry. Oh, that wasn't updated. My apologies. But he is going to show us how the use of DGRP can map gene interaction, gene networks that are mediating autism-like behaviors.
Thanks. Tennessee. So I want to thank the organizers for having me here. I get a chance to talk about a project that's been ongoing for a few years in my lab, just sort of in the background, and using a resource that I really appreciate, the DGRP collection, which I'll talk about a little bit. And the primary focus of my lab is autism. And, you know, as some of you probably know, autism is a genetically complex disorder. And I think that these approaches using the DGRP collection, for example, is probably a better way to get at it than starting with humans. When you have monogenic disease, it's a little easier to go that direction. But in this case, I'm going to show you some data where we use the DGRP to identify some, in fact, a new gene that I think is going to be relevant. So as I mentioned, there are a lot of genes involved in autism. This is not an updated slide. There's somewhere around 800 autism-associated genes now, which makes it very complicated to decide which gene to work on. And in fact, these genes probably interact most likely, and so you have to identify genes that interact with other genes, which is a much more complex problem than a single gene disorder. So I was trained in human genetics, so I like to think about phenotypes first. And in this case, some of you may not be familiar with how autism is actually phenotyped in a research study. And according to the DSM-4 criteria, which I'm going to show you, it can be separated into three classes, three different behaviors that you want to look at together. And those would be social interaction and communication in a social context, which is an important point, not just communication, but social communication, and restricted or repetitive interests, which is sort of the classic feature of autism that probably most people are familiar with. The communication and social interaction have been sort of fused in more recent definitions of autism, and it really is social communication that we kind of want to look at. So together, these three make up what we call the autism phenotype in humans. And what we wanted to do was to find a way to look at autism using Drosophila. So what we did was we looked for behaviors that were similar or autism-like in flies. And in fact, if you think about it, there's a recent study showing that you can look at social space. In fact, some people have talked about it at this meeting. You can put some flies in a triangle and you can see how far apart they settle over time, over about an hour. And they have a defined social space, and mutants in the autism spectrum actually have inappropriate social spacing in this assay. We can also use mating as a measure of both expressive and receptive communication during social interaction. And finally, grooming assays. Again, people have shown that grooming assays done in, for example, Fragile X mutants and flies, they show a robust phenotype, and there's a restricted repetitive interest type phenotype in grooming behavior. So what we did is we looked at all of these behaviors together, and what we're really trying to do is fuse them in what we call the F-ADOS, or the FLY ADOS, which stands for the Autism Diagnostic Observation Schedule, which is what's used in people to diagnose autism. So, as I mentioned, we use the Drosophila Genetic Reference Panel for these studies, and for those of you who are not familiar with this panel, it was developed by Trudy McKay, and it's a really interesting collection of flies. There's 205 deep-sequenced, wild-caught female isogenic lines. They were back across 20 generations, which eliminated variation within each line, and they represent more than 4.6 million SNPs, which gives them a lot of genetic power. And the genetic variation across the lines reflects variation in the natural population where these lines came from. In fact, the lines are variable across traits that are tested, and almost every trait that's been tested, these 200 lines are variable. And in fact, they've been tested for a lot of things. So at this point, a lot of the focus has been on traits that may be normal behavioral traits or things that are not related to human disease. But a few papers have come out recently using these lines to look for suppressors and enhancers of phenotypes related to disease. And what we wanted to do was use them in our F8OS and actually see if we could fuse together those behaviors and find major alleles and probably minor contributions to phenotypes by using this collection. So why are we using them? You know, although 30% of ASD cases can be mapped to genomic lesions or mutations, about 70% of cases, or what we call idiopathic autism cases, are due probably to combinatorial effects and maybe environmental effects as well. So there's a clear genetic etiology, however, because if you look at twin studies, and these are sort of classic twin studies, they show that 90% of these individuals are concordant for the phenotype, but if you look at siblings, it's only about 5 to 10%. So it's not one gene that they have to inherit, but many genes to have this phenotype in common. So the DGRP allows us to identify combinations of genes. We can identify individual genes or SNPs in or around those genes that are associated with the phenotypes, and then go back in flies and combine those genes that we think are controlling these behaviors. So as I mentioned, you know, the collection has substantial genetic power and probably a lot more power than other systems, which is what attracted us to using the DGRP lines. So how are we doing the experiments? For social communication, we're using mating latency, and we put the DGRP line either in males or in females to look at either expressive or receptive communication. So you're talking about expressive receptive communication during social interaction. For social space, we separated males and females. We looked at males in the triangles, we looked at females in the triangles, and I'll show you some data that came out of that later. And then for repetitive restricted behavior, essentially we observe the flies grooming over a 5-minute interval, and we look at the amount of grooming during that period. We looked at 40 DGRP lines. We did our best to randomly pick 40 DGRP lines, and all of the behaviors that we tested showed normal distribution across those lines, and each measure was performed 3 times per line. So when we had all of our data, our behavior data, and our measures, we plugged them into the GWAS tool that's available through NC State for this collection, and out pops magical heat maps, right? So I'll walk you through this data, and for those of you familiar with the collection, you might notice that there's a lot of color on my heat maps. We haven't looked at 200 lines, we looked at 40 lines. However, I would like to stress that, you know, we're interested in both the major contributions and the minor contributions. So I think there's some meat here that we're looking at, and we're gonna see in the future here if we are. So if you notice that for female receptive language and male expressive language, there's a couple interesting hits that pop up. Some of them are autism-associated genes you might be familiar with, neuroligin and neurexin. But one that came up that came to our attention was this SFL gene, and it came up in both. And in fact, SFL came up again in social space, and it came up again in grooming behavior, although not a very large hit. Indeed, there was a hit that was significant. So that got us interested a little bit in this SFL gene. And if you look overall at the data we've collected so far, in fact, there's a little variability in how many hits we get. We get a few more hits for grooming behavior, but we only get that single gene that hits all of these categories that fits our criteria at this point for what we're trying to investigate. There is another gene that is sort of uncharacterized in flies that overlaps with social space and grooming. I'm not going to talk about that gene. So what is SFL? Sulfateless. It regulates heparan sulfate residues in the nervous system. It's been shown at the neuromuscular junction to alter synaptic function in flies. And in humans, heparan sulfate modifications are critical for cell migration and neural circuit development. So it's kind of an interesting hit because a lot of people talk about autism as a synaptic disorder and a cell-cell communication type of disorder. So this may be a critical gene. So in addition to the data that I showed you with the association studies, we actually dug a little bit into some recent data that was published on gene expression in these lines. And what we were really looking for here is on the bottom, we have the number of each of the lines that we used in our study, each of the DGRPs. And then on this axis, you can see the gene names, although you can't read them. And the goal was to try and use some recently published gene expression data to find patterns where maybe the gene expression was related to those SNPs in those genes. We did not find that for female receptive communication. We didn't really find that for male expressive communication. We got a little bit excited when we looked at social space. And then it turns out, when we looked a little deeper, that this was the female experiment and this was the male experiment. So there is a difference in gene expression, and it's sex-specific for these particular genes in these particular lines. So it kind of fooled us into thinking we had found something. So really, the focus is on sulfateless right now, and we found a single gene that crosses all of our behaviors that we looked at in the 40 DGRP lines. And we did not find a correlation between gene expression and these behaviors, but we've only done 40 lines. Maybe if we do more lines, we'll be able to see such a correlation. There were other autism-associated genes that we identified, including neurexin and neuroligin 2. So that was good. That meant that we might be on the right track. And then we found a gene called KIRE, which actually is associated with intellectual disability in humans. So again, it looks like we're finding things that kind of make sense to us. So the future, really, the first thing we need to do is to validate that sulfateless is involved at all in any of these behaviors. So we're in the process of doing RNAi knockdown and overexpression studies in neurons. And we'd also like to validate some of those other hits and our goal is to complete at least 100 DGRP lines, because we think at that point we can do some Z-score analysis across these behaviors and possibly identify multiple genes contributing to the phenotypes. So with that, I'd like to acknowledge some of the people that helped. Obviously, Trudy McKay and Robert Onhold for creating the DGRP, and my collaborator Janice O'Donnell for coming up with the F8OS idea with me over some mojitos at a fly meeting, and Kevin Hope, my graduate student, who's sort of been directing this project with multiple undergraduates, because it's just something we kind of do in the background. And it's been funded entirely through some department funds because I can't seem to get traction for autism and flies for some reason. And with that, I'll take any questions. I think I got like a minute or two.
I have a question. So I assume that you're looking at social spacing using same sex,
Yes.
which is male. So flies are not exactly social animals. So
Right.
how could this social spacing represent one of the phenotypes in social communication?
So what we're looking for is really inappropriate social spacing. So the kind of social spacing we want to see is do the males or do the flies, male or female, get closer or further apart than they typically would do if you look at wild type animals. And it's kind of a subtle thing to look at, you're right, but they're not exactly communicating. It's more like, when you walk into a room and somebody stands right here at a party and you feel a little awkward about it and you kind of walk a little further away. Well, these flies do that, right? They have a distance that they're comfortable with sociably, and they violate that distance. Some particular lines will violate that social distance.
I have one question. Given the male preponderance for autism, that you have a 4-to-1 ratio at least, when you look at the genes in your male, kind of expressive behavioral deficits, did you see kind of an interesting overlap with your other modalities in the F8OS?
I'd have to say I don't think we have quite enough data to answer that question yet. But it's a good point. And there certainly is a prevalence difference, which may have more to do with where the genes land in humans than anything else. So, I don't know.
Okay, great. Thank you so much. This is very interesting.